All right, good morning, everybody. Thanks for being here on Monday morning. It's good to see almost everybody here. Who's missing? Matt, Rand, and Abby. I think otherwise, and Sophie. Okay. Cool. I'm glad everyone's car batteries worked. I hope you had a good weekend, enjoyed some sunshine. We're gonna hop right into it today, picking up where we left off, talking about the nervous system. Before we do, a couple of quick reminders, announcements. There is a talk tomorrow, Tuesday afternoon at 4:10 in the Science Center. We have a special guest coming. Check it out. It's going to be great. Tell your friends. Also, I presented this last week, but I'm recruiting students to work in the lab, doing research projects this spring and especially next summer and even beyond. If you're interested, let me know. There's information about the lab website here, and the job is posted on Handshake. All right, today's agenda, we're going to jump right back into the nervous system and talk about support cells. Actually, that's not entirely accurate. We have a little more to talk about with neurons, and then we're going to get into support cells. And I have a human anatomy in the news segment for you. And then we're going to continue talking about the nervous system, see how far we get. And we'll wrap things up. I have some scheduling updates and reminders, and then we have a ticket out, and I have a super surprise for you at the end of class as well. So stay tuned. Big surprise coming for the end of class. As you work through the nervous system, don't forget you've got a study guide, a Google Doc, Google Slides file that has diagrams and resources for you as we work through. So let's go back, let's hop right back into it and talk about neurons. And I want to specifically just talk for a few minutes about the structural types of neurons. So just as a quick reminder of our anatomy, the cellular anatomy of neurons. If we look here, I want to make sure you remember the kind of three major anatomical parts of a neuron are the cell body, which is this bigger bulbous portion. It's filled with cytoplasm. There you'll find most of the organelles as well as the nucleus. Coming off of the cell body, we have these dendrites, and then usually, but not always, we have one longer extension called the axon. Now, there were some other components of the anatomy I talked about on Friday, but those are the three main big ones. By way of reminder, remember that signals come to the cell body via the dendrites, and then from the cell body, a signal is sent down the axon. So remember, away from the cell body, axon, both start with the letter A. Now, having said that generalized structure, neurons can take on a couple different forms with those basic components arranged slightly differently. The first is what's called a multipolar axon. This is the first one you see over on the left there, where there's lots and lots of dendrites, and they all connect directly to that cell body, and then there's one axon leading away from that cell body. And that is called a multipolar, and most of the neurons in the central nervous system are this type of neuron. And this is what you most commonly see in textbooks and things like that when you see a diagram of a neuron. Now, there's also a kind of neuron called the bipolar, which looks like this. And basically what you have here is there's these branches of dendrites that connect to one long dendrite leading to the cell body, and then one axon leading away. And so these kinds of cell neurons aren't very common. They are found associated primarily with some sensory organs, eye, nose, and ear, for example. And basically, all you'll notice here is that the cell body is kind of moved down so that it has this long dendrite on one side and a long axon on the other, hence the name bipolar, because it essentially looks like it has two long branches coming off either side. Important to note, those branches are not the same, that one is a dendrite and one is an axon. Nonetheless, kind of when you just look at the structure anatomically, it looks like it's pretty similar on either side. Finally, we have something called the unipolar that you see here. This is when the cell body kind of comes off of the axon in the middle. And so you have the dendrites down here, these kind of what's called these receptive endings. And then you have a first part of the axon, you have the cell body off of that, and then you have the rest of the axon there. And so we call this unipolar or pseudo-unipolar. The reason it's pseudo-unipolar is because it's actually a bipolar cell and the cell body has just kind of moved off of it. So technically it's a kind of bipolar neuron, but we're just going to call it unipolar. Just know that sometimes it's called pseudo-unipolar. This one, again, is not super common. It's associated primarily with sensory neurons in the peripheral nervous system. So this is things like the sensory neurons associated with your integument to provide sense of touch or temperature and things like that. So 3 basic structures of your neurons: multipolar, most common, we see here; bipolar, we see here; and then unipolar or pseudo-unipolar, you see here, where the cell body it just kind of comes off this little branch off of the axon. So given these 3 general types of neurons, there's a couple different places where we find them in the body. And I want to talk in a little more detail about how these neurons are structured in the body, where the cell body and axons are in the different types of neurons. Now, what's important to note is that in some cases, this axon can be super, super long and extend, in some cases, let's say from your spinal cord up here in your lower back, all the way down to your toe. So these can be like a meter long, these axons of a single cell, which I think is pretty cool. So let's talk about okay, so this is a complicated slide. I'm going to walk you through it. Before I walk you through this one, let me pause here and see what questions you have so far. I know it's a Monday morning. I'm just going for it, though. I'm just diving in. Go ahead, Zoe.
So are the multipolar ones more common because they can be more thinking with the difference, or is there only this one?
Good question. Is the multipolar one more common because it can receive more information? I'm just trying to think about the best way to answer that. So here's what I'd say is because these cells can receive information from a whole bunch of different neurons, they're going to be most common in the central nervous system because there we have information being integrated. So you have all of these different neurons giving information to this single neuron here, and then that neuron sending information out. So in that sense, yeah, I never thought about it. But that makes a lot of sense because that's where all of these signals are kind of coming together and crossing, and that information is being integrated in the brain, especially. Whereas in the peripheral nervous system, generally we have a signal that's just traveling a long distance to the central nervous system. And so you don't need as much information integrated. Yeah, it's a great way to think about it. Thanks, Zoe. Good, good. Other questions? Cool. All right, so let me walk you through this slide a little bit. I want to functionally classify neurons. Remember, neurons are functionally classified based on the direction of the impulse relative to the central nervous system. So sensory neurons are bringing information to the central nervous system. And except for those neurons associated with the special senses that are bipolar, most of these are unipolar. And what they are is, what's important to note here is you can see the actual cell extends both from the peripheral nervous system into the central nervous system. So these sensory neurons, we can't say that's in the central nervous system, that's in the peripheral nervous system, because they span both, right? They receive information here in the peripheral nervous system that's sent down. Their cell body is in the peripheral nervous system, but that information is ultimately delivered via, remember these, the axon, the terminal branches of the axon are here, and we have our neurotransmitters coming out. That synapse is in the central nervous system. And so we can see this in a couple different diagrams here. It's basically the same thing, just dividing this into the part of the cell that's in the peripheral nervous system and the part that's in the central nervous system. Importantly, note that the cell body is in the peripheral nervous system. And we're going to see where those cell bodies group up in places called ganglia that I mentioned a little bit on Friday. Now, I have a couple examples also of bipolar cells that are associated with the special senses. And you can see what these look like here. Essentially, the cell body is just kind of in the middle of this long branch of the axon and the dendrite here. And it doesn't come off on a little side branch like it does for a unipolar or pseudo-unipolar cell. So those are our sensory neurons delivering information to the central nervous system. They get information from the periphery. They deliver that information to the central nervous system. Now, of course, the other kind of neuron, we have sensory neurons, and what's the other one? Begins with a mo and ends with a ter. Motor. Excellent. You guys are good. Okay. So motor neurons, of course, are neurons that are delivering information in the opposite direction. They're delivering information from the central nervous system to the periphery of the body, and specifically muscles and glands, where they tell those muscles and glands to do something. These neurons are generally or always, I'm going to say they're always, with few exceptions, they are multipolar, meaning that they look like the diagram you see here. Their cell body is within the central nervous system, with a couple of exceptions. And they then the axons extend into the peripheral nervous system, and they interface with muscles and glands here. So the motor neurons have their cell body in the central nervous system. So let me just put some information here as just a summary. The motor neurons deliver information from the CNS. I'm just going to say to the body, meaning generally across the body, and the cell body is in the central nervous system for motor neurons. Sensory neurons, on the other hand, line here, they deliver information from the body, the external or internal environment, to the central nervous system, and they have their cell body in the peripheral nervous system. Make sure this shows up on the camera there. There you go. So that's the two big takeaways that I want you to know here. I also, actually, no, there's one more too. One more takeaway as well, is that motor neurons are generally multipolar, and sensory neurons are generally unipolar. And then, of course, some of the special senses are bipolar. So that's the big summary points that I want you to take away from motor and sensory neurons. Note that both types of neurons span the peripheral and central nervous system, because they're providing information across those aspects of the system. Let me pause there, see what questions you have, and then we'll talk about the last type of neuron. Here I can get this on camera. Yeah, that looks pretty good. Cool. So there is one more type of neuron, and that is what's called an interneuron. These are found exclusively in the central nervous system, and the vast majority of your neurons in your body are these kinds of interneurons. Now, kind of going back to Zoe's question, because these neurons are in the central nervous system, they're often shorter, and they are multipolar with lots of like crazy arrangements of their dendrites. And you see some examples here. This is what's called a Purkinje cell in your cerebellum. We'll talk about the cerebellum next week. This is called a pyramidal cell. We'll talk about those guys very briefly as well. I don't need you to know the details of what these different kinds of branches look like. I just want you to recognize that an interneuron has this crazy arrangement of dendrites, and that they are found in the central nervous system. So going back to what Zoe asked earlier, the reason for this is because these guys are integrating information. So they're getting all of this information from different axons at the same time. Hence these complex arrangements of dendrites to bring that information. And the percentage is something, I mean, it's 99.98% of the neurons in your body are these kinds of neurons. Now, what's important to note is that calculation is based on number of cells. These cells are often very, very small compared to your sensory neurons, which, as I said, can extend longer distances. So if we did this by weight of cell or something like that, it would give you a different number where it wouldn't be quite such a high percentage. But if we look at the actual number of neurons, almost all of them are these interneurons. So in summary, we have these different structures of neurons. We have multipolar cells that look like this, that are, for example, a motor neuron. Then we have these unipolar cells, which are associated with, which are sensory neurons. Special sensory neurons are bipolar. And then we have a whole bunch of different kinds of wackadoodle multipolar arrangements that we can find in the interneurons. So you should be able to look at a diagram and tell me if a neuron is bipolar, unipolar, multipolar, and then if it is multipolar, you should be able to distinguish the shape between a motor neuron that you see here, kind of this classic neuron shape, and something like this that we would find as an interneuron. And that's all I'm going to say about the cellular anatomy of neurons. That's all we're going to cover in this class. So to remind you, nervous tissue has two main cell types. The first are neurons. These are the specialized cells that transmit information via electrical signals known as action potentials. We've just talked about those guys. The other type of cell, in fact, the majority of cells in the nervous system are what's called support cells, or in the central nervous system, we call these neuroglia or neuroglial cells. These are cells that don't transmit information, but do a whole bunch of other stuff to ensure that your neurons can transmit information. So what we're going to do now is transition and talk about these support cells in a little more detail. So as I mentioned, that neuroglia, or also known as neuroglial cells or just glial cells, are supporting cells in the nervous system that provide this scaffolding and kind of physiological support for the neurons of the central nervous system. These cells cover, so meaning they kind of literally wrap around the non-synaptic parts of neurons. So they help insulate that electrical signal in the exact same manner as the wires in these walls are covered in plastic. And I'll talk a little bit more about how that works in just a second. But I want you to know that's one of the important roles of glial cells. Most of these glial cells have these branching processes in a way that looks structurally similar to neurons a little bit, but they're often smaller and these processes don't extend as far. So the takeaway here is that most of these glial cells have the cell body with stuff coming off of them. As I mentioned, the majority of your nervous system cells are glial cells by a count of about 10 to 1. So they make up like half the mass of your brain are these supporting cells. And finally, unlike neurons, glial cells divide and reproduce and produce more glial cells throughout life. And so this, because these cells are undergoing mitosis and dividing, this can lead to the possibility of tumors. Remember, a cancerous tumor is a cell that's basically not doing what it's told and going rogue and just trying to produce as much as itself as possible without regard for what the effects are on the rest of the body. And so when you hear about brain tumors, for example, a glioma, most of the time it's not the neurons that are cancerous, rather it is these glial cells. Because remember, neurons don't really divide. Generally speaking, most often speaking, they don't divide in adults. And so there's not a chance for those cells to become cancerous because they're not dividing in the first place. So when you hear about brain tumors and brain cancers, it's a cancer of one of these types of glial cells. And we're going to talk about the 4 types of glial cells in the central nervous system, and then 2 types of glial cells in the peripheral nervous system. And then you will all receive your certificate in glial cell mastery that you can show off to your friends and family when they ask you what you did this semester. You can be like, booyah, glial cells, what's up? All right, let's talk about these 4 cell types. So first, I want to focus on the 4 types of glial cells in the central nervous system. And as I said, there's 4 of them. The first are what's called astrocytes. These are the most abundant, and they have an active role in neuron activity. And specifically, they're important for regulating the rate of neurotransmitter reuptake. So we're not going to get into the physiology of this. Two important things I want you to know about astrocytes is one is they are they're structurally they have a cell body and then all of these processes coming out that are wrapping around not only the neurons but the blood vessels found throughout nervous tissue. And they're important for regulating neurotransmitters. So if you recall how signals are transmitted across that synapse, we have these little molecules called neurotransmitters that are released by those terminal boutons of an axon. They travel across the cytoplasm in that synapse, and then they're picked up by receptors in the dendrites of the next neuron. Now, what needs to happen is if those neurotransmitters aren't picked up, something needs to regulate them and pick them up and bring them back into the neuron so they can be reused. We need to make sure there's not an excess of these neurotransmitters floating around and setting all kinds of signals that we don't want sent. And it's these astrocytes that are responsible for regulating that. Again, I'm not going to get into tons of detail about it. I don't need to worry about that for this class. Just know that it's an important glial cell in terms of regulation of neurotransmitters. Your second type of glial cell in the central nervous system is called microglia. These are immune cells that are phagocytic, excuse me, let me say that again, phagocytic, and they wrap around and engulf invading microbes or dead cells of your own. And they then basically clean, they keep the central nervous system clean of invaders. You can think of it that way. They are not terribly abundant and they're pretty small structurally. And what's important here, as I mentioned, they're immune cells. They're derived from this kind of immune cell called a monocyte that migrates to the central nervous system very, very early in development before birth. So microglia, you can think of these as the immune cells of the central nervous system. Yeah. Okay, so they migrate there early in development. I believe I have to double check this, but I believe the monocytes early in development migrate to the central nervous system where they become microglia, and then those microglia kind of divide to produce microglia. Let me verify that. I'm almost positive that's the way it works, but I'll check that. And that's a great question. Okay, number 3 is what's called an ependymal cell. These are epithelial cells, actually, which is pretty cool. And they form a selectively permeable membrane lining the central cavity of the central nervous system. And they basically have these little cilia that help circulate cerebrospinal. Now, we haven't talked about the central cavity yet. We haven't talked about cerebrospinal fluid yet. We will in the coming weeks. Right now, just know that cerebrospinal fluid is this specialized fluid that basically encapsulates your central nervous system. So it's found around your brain and actually down the middle of your central nervous system, there's a canal called the central canal or central cavity. And these cells just keep that cerebrospinal fluid circulating. We'll cover that in a little more detail when we get to the brain anatomy and your lab in a couple weeks. But for now, I just want you to know that these ependymal cells are kind of epithelial cell forming the lining around this cavity where we have the cerebrospinal fluid. Oh, wait, I put this in the wrong place. Oops. All right, I'm going to skip ahead to number 4 here. These are oligodendrocytes. These are cells that produce the insulation that wraps around the axons of your neurons in the central nervous system. And so they have these processes that reach out basically, and they are formed of a structure called myelin. That's a kind of insulating protein. So this is, as I mentioned earlier, basically when you have a wire, like a copper wire with plastic around it, this is the insulating plastic. This keeps that signal moving quickly and efficiently where it should. If you've taken 251 with me, or if you've taken 325 with Dr. Penwis, you know more about action potentials and how this myelin sheath helps these signals move in this kind of jumping fashion that allows them to move faster. For us, I just want you to know right now that it's these oligodendrocytes, which are a type of glial cell that produce this insulating myelin around the axons of the neurons in your central nervous system. Interestingly, I'll get to you 1 second, Morgan. Multiple sclerosis is a pretty dreadful disease that most of you have heard of, I'm sure. What it is an autoimmune disease where the immune system decides it wants to attack this myelin. And so what happens then is that myelin degrades and it disrupts the integrity of those electrical signals sent by the neurons. So it can lead to things like inability to control muscles and things like that, a variety of nervous system and neuron signal related trouble, but it's actually an autoimmune disease where that myelin gets degraded. Morgan, question.
So the bipolar, like they didn't have the island sheet in the picture.
Good call.
Does that make them more susceptible to like sellers?
Yes, you're talking about like this side here, where you have the bipolar ones right here.
Yeah.
Yeah, great. I'm really glad you noticed it, Morgan. No, it's just a crappy picture.
Okay.
And crappy in the sense that I'm showing you the myelin around the axons and these multipolar cells. But for whatever reason, it doesn't have it. But what you would see here is you'd have myelin generally around the axon here or the axon here. So it's not going to be around the dendrites, only around the axon. So it generally will be. Although it's important to note that not all interneurons are myelinated. Sometimes they're so short and they travel such short distances, the myelin wouldn't really help much. And it's better to have more of these packed into a smaller space. So not all nervous tissue is myelinated, but those bipolar cells here generally will be myelinated around the axon. Great. I'm really glad you noticed that. That's a really good point. Oligodendrocytes, anything else? All right, let's talk about two types of glial cells in the peripheral nervous system, or support cells in the peripheral nervous system. The first is called a satellite cell. You can see a diagram here of the axon, an axon in the peripheral nervous system. And you can see the satellite cell essentially surrounds the cell body and kind of supports the cell body. So these cell bodies are grouped in those ganglia, and then each of the cell bodies is wrapped in these satellite cells. And then we have the Schwann cells, which are basically oligodendrocytes, but in the peripheral nervous system. They're also known as neurolemmacytes, and they form this myelin sheath around the axons of the neurons of the peripheral nervous system. So Schwann cells and oligodendrocytes, same thing, different place, basically. There's some other smaller differences, but it's not, for our purposes, that's a good way to think of it. They both produce that insulating myelin sheath. So that is the 6 types of support cells that we'll cover. Let me just see what I have here. I've got a little more. I want to talk about myelin sheath in just a little more detail, but I want to do a few other things before we get to that. All right, I'm going to break things up. I'm going to go back to human anatomy in the news. So this is kind of cool. A study was looking at brain activity after death. So kind of a fascinating study to think about what is happening in your body on a cellular level after you die. And there's actually a lot of research into this kind of thing long-term in terms of decomposition and things like that. This study was looking at very short-term responses after death. And what happens is when you look at the activity of different genes in these cells over the course of 24 hours, different genes responded differently. So they're not all doing the same thing, which isn't surprising, right? But what is kind of interesting is that housekeeping genes maintain their normal function, meaning that even though the neurons weren't really doing their thing at all of these other support cells were active in trying to keep things going. And so after 12 hours, these neuronal cells reduce their activity, but genes that were linked to glial cells, so these support cells, actually increased and continued increasing for up to 24 hours. So after death, your neurons decrease activity, but all of these other support cells, I should say all, a lot of these other support cells are actually increasing activity, and that activity is spiking. Any guesses why that might be the case, based on what you know of these two cell types and their functional roles? Go ahead, Gia, what do you think? Talk a little bit when you say rephrase that sentence, but tell us when you say they're trying to restore them. Tell us what the they and the them are. Uh-huh. Mm-hmm. 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 Yeah, that's exactly what the researchers think. Nice work. So they're likely responding to the injury and inflammation in the brain when it's deprived of oxygen. So all of these processes shut down in the neurons. And remember, these support cells, their job is to keep those neurons working. And all of a sudden, the neurons are totally crashing. So these glial cells are like, oh shit, like we got to get on this. We got to keep these neurons going. And so they're ramping up their activity as much as they can, trying to combat that, not knowing that it's ultimately a losing battle, of course. But yeah, that's exactly what the researchers think is going on. This article also has, I have links to this in your class program for later in the week. It was also in your class program for today, but we didn't get to it on Friday. It has a great quote from one of the researchers that I love. It says, the results don't prove that zombies are theoretically possible. So just in case you wanted to misinterpret this, I know Halloween's coming up. It says pretty unequivocally here that that's not the case. So a cool study that came out a couple years ago, directly relevant to what we're talking about here. Go ahead, Julie.
Mm-hmm. If something in your body is still working,
Mm-hmm. Mm-hmm. Right. That's a great philosophical question. And here's how, without getting into the weeds and how we define life and death, right? I think what we want to think about is that our concept of our existence is really tied to neurons and this brain activity, right? And at a certain point, if those neurons stop firing, then there's no turning back. Everything else goes downhill. So even if these housekeeping cells are doing their jobs, or, you know, any number of cells could remain functional for days after you die, even, you know, let's say the cells in your digestive tract, they maybe have nutrients that they need because there's food in your digestive tract, continue working for a few days afterwards. We don't really associate our consciousness or life with the cells of our digestive tract or like the immune cells that are in our brain even. So yeah, that's a good question though. Like at what point do we consider it? And it gets more complicated, you know, if you think about different neurons degrading at different rates and things like that, or someone who's quote unquote brain dead, where, you know, your higher or higher level cognition and your ability to like think and interact with the world is gone, but the parts of your brain that control your breathing and your heart rate still work. Like that gets really complicated, right? And there's big ethical and legal questions around like what to do with people in that state or whatever. Yeah. Yeah, it's a great question. I like thinking about that stuff. Cool. All right. So what I want to do now is a little activity. And you're going to submit this with your ticket out. Oh, I said 2 points, but ticket out today is actually going to be 3 points. So in your notes, draw a grid that looks like this, but make it bigger. I should have made it like, say, take a whole piece of paper, or not like half a paper maybe, and divide it into quadrants. And what I'd like you to do is, from memory, spend 1 minute and list the 4 types of glial cells in the central nervous system and just brainstorm, that's a good term here, whatever you can about them without consulting your notes. And then after a minute, we'll go and consult your notes. So just try your best off the top of your head. All right, what are the names of those 4 cells? What are their functional roles? What do they look like? All right, now go ahead and look at your notes and make any additions or amendments that you'd like to that. And now you have a little handy study guide of the 4 types of glial cells in the central nervous system. I see lots of people writing. I'll give you another minute. Let's see what I want to cover. All right, I have one more short thing I want to talk about today, and that is this myelin sheath I mentioned. Remember that myelin sheath is this insulating protein that's wrapped around the axons of neurons. In the central nervous system, it's produced by oligodendrocytes. In the peripheral nervous system, it's produced by Schwann cells. And well, I've been calling it a protein. I guess technically it's a lipoprotein, so it contains fatty molecules as well. And as I just mentioned, it's formed by oligodendrocytes and Schwann cells. And what you can see, I don't know if you can see it well in this diagram here, but this is a cross section of an axon. So a transverse section looking down the end of an axon. And you can see here this myelin wrapped around the axon to provide insulation. And specifically, this myelin performs 3 important functions. It prevents leakage of electrical current, so it keeps that electrical current moving where it should. It actually allows that current to move faster, so you can transmit information over long distances quicker. And it makes this, this propagation more energy efficient as well. So it does all the things. It keeps the signal going where it should, it keeps it moving quickly, and it allows it to do so efficiently. So especially when these signals are traveling long distances, this myelin is essential for that operation. And I think, well, I'll just mention a little bit more here on the next slide. So from an evolutionary perspective, if we have a signal that we want to transmit long distances quickly, well, there's a couple different ways we can do that. One is through this insulation and using this myelin to allow signals to kind of jump down the line and move faster. Another way to do that is simply to make your axons bigger. And from an evolutionary perspective, some of us vertebrate, or some of us animals have gone one route with these insulated axons, but others, such as giant squid, they have to transmit information long distances. Instead of having myelin, however, they just have really big axons that allow, because of the properties of conductance of electricity, if you have a bigger diameter, a signal can travel faster. And so they have axons that are like, visible to the naked eye, several millimeters in diameter. So just as a quick aside, you don't need to know all the details on this slide. Actually, I don't like the way I presented this slide at all. I should have just had a picture of a giant squid. All I want to share with you is that if we don't have that insulation on our axons and we want that signal to transmit faster, the alternative is to have a much thicker axon. I do have one more slide I want to show you here. And that is a little bit about what happens when these neurons are damaged. If a cell body of a neuron is damaged, like that's it. That neuron is done for, right? But if the axon is damaged, there is a process through which that axon can be repaired. And I kind of walk through what happens here. But basically, the Schwann cells in the peripheral nervous system can reform the tube, a tube, and the axons can regenerate within that tube. And there's macrophages help clean up the debris and things like that. And then you can develop a new axon with a new myelin sheath around it. Now, this process is complicated and takes a while. And importantly, it's specific to the peripheral nervous system. So I said that the Schwann cells can help regenerate this structure. Remember, Schwann cells are in the peripheral nervous system. Your oligodendrocytes in the central nervous system don't do this. And that's why these cells can't repair themselves in the central nervous system, only in the peripheral nervous system. So there is a section in the book that talks about this. It's a part of your reading that gets into a little more detail. I don't need you to know all of the details about how this tube is regenerated. Just know that it happens because of the actions of the Schwann cells, which are only in the peripheral nervous system. And that's all I'm going to say about the regeneration. All right, we're in good shape here. I want to bring us to the end. What questions do you have, actually, before I do that? Should have a few announcements and reminders for you and a ticket out. Okay, let's get to this. We'll finish all of this up later in the week. So our upcoming schedule, important reminders. Some of you have asked me about this, so I want to make this clear. Lecture quiz number 4 will be a week from today in class, closed book quiz. So it'll be a similar format to the last quiz. A week from today, you'll have 20 minutes. It's going to cover the skull and central nervous system through what we talked about today. A week after that, you're going to have lecture exam number 3. And this is going to be open like the last exam for 2 days, a Sunday and a Monday. We won't have class on Monday. It's in Blackboard and open book. So I was debating about the format for this upcoming quiz and exam, and I decided that a little bit you'd hate me for the closed book quiz, but then I thought I'd make it up to you with an open book exam. So we've got those 2 things coming up. This information is in your class program as well. So quiz a week from today in class, exam the following Monday. We won't have class, it'll be open in Blackboard. Questions on that? Okay, just as a reminder, lab practical is tomorrow. Remember that the morning lab is going to start at 10 a.m. so I can get things set up. Remember, the lab is open tonight. Well, it'll be open regular hours tonight, but starting tomorrow at about 6 a.m., it's going to close. I'll be in probably about 6:30 tomorrow and use the time between 6:30 and 10 to get it set up. So you can't come in and study during those hours. I know a lot of you want to come in at 6:30 tomorrow, but nope. Go ahead, Kelly. The lecture quiz is next Monday. Yeah, so a week from today. And then lab practical is tomorrow. I see confused faces. What question can I answer? Like in lab? Yeah. Okay. Yeah. I wanted to say, like, don't worry about the quiz. Focus on your lab practical. The quiz will be sometime in the future after the lab practical. And that sometime in the future is a week from today. Cool. Here's the other thing. Here's the special surprise announcement. We've been cranking with this class. We're in good shape with our curriculum. You've got a lab practical. Hopefully you've all been putting in a good amount of time and energy to prepare for that. We're not going to have class Wednesday. So take Wednesday, catch up, sleep in, do what you want to do, take a little bit of time off. I was worried. I was like, maybe students don't want to have canceled class. Maybe they'll be upset if we can't. It's Abby's birthday. So happy birthday. So your assignment for Wednesday is to wish Abby a happy birthday. No, it's been a crazy week. I know it's advising week 2, which, you're getting your schedule set, just a lot going on. Take a breather after your practical tomorrow. Note that you do have a class program for Friday. So there's some readings and other, a video for Friday, a short, like one of the crash course A&P videos. I also have information and more details about your lecture, quiz, and exam on the practical. But it takes some, so I just want to emphasize that there's still some things to do for the next class on Friday, but we won't have class on Friday, or excuse me, we won't have class on Wednesday. Take a breather for yourself and we'll get back to it Friday. No one's too mad about that, I'm sensing. Okay, cool. Let's wrap things up here. Just a couple quick reminders. Danny will have a tutoring session this afternoon. I highly recommend going for preparation for your practical. I have open hours today. Note, with apologies, I have another conflicting meeting scheduled, just happened to be the way it is, but I will be in that room from 1 to 2 today. So come by with any questions. Otherwise, a ticket out. I realize that your class points aren't adding up fast enough. So I'm making a lot of these in-class tickets out and things like that, 3 points instead of 2. So we can get to the total 100 for the class for the semester. We're a little bit behind where I want us to be. So stuff is going to be worth more points moving forward. Otherwise, let me know if you have any questions. I will see you in open hours this afternoon. If not, I will see you tomorrow. If you're in the morning session at 10, if you're in the afternoon session, regular time, 1:10 p.m.